So we all know that the composition of nitrogen, molecular nitrogen in atmosphere is 78%. Okay, they exist in many forms in the biosphere, and these forms undergo transformation. They change from one form to another. They change from one physical form to another, and we will today study about the three major transformation, which is the biological nitrogen fixation, ammonification, and nitrification. So biological nitrogen fixation is what we will do today. What is biological nitrogen fixation? So the conversion of nitrogen, which is molecular nitrogen, into ammonia by some bacteria. Since the word biological is there, by bacteria is known as biological nitrogen fixation. So here the end product is ammonia, and this is the first stable product of biological nitrogen fixation. Okay, and all the bacteria which are involved here, they all are prokaryotes. Okay, we will study about the details of this bacteria in after few slides. So ammonification is what? These ammonia which is formed like molecular nitrogen to ammonia. Now the storage of this ammonia is very toxic to the living organism. So what happens? This ammonia is converted to the organic form. Ammonia is the inorganic form. This will be converted into the organic form. How it will be converted? It will be converted into amides and urides, which will be incorporated into the organic molecule, okay, which we will study later. Now, today biological nitrogen fixation, the association is between the bacteria, which is here the rhizobium, and so the association is between the bacteria and the legume plants. So we will study about the bacteria and the legume plant separately. First, let's study about the legume plant. So this bacteria and legume plants, they both exist in the symbiotic association. So what is symbiotic association? It is that they both help each other in the survival. Okay. Symbiosis term, I think it's familiar with you all. Like they both help each other in their survival. It's a kind of mutual relationship. They form a symbiotic association. So let's start with what are legume plants. So example of legume plants include what? Soybeans, peanuts, alfalfa, clover. So all the dal family of plants. The pulses plants include the legume plants. These plants live in the symbiotic association with the rhizobium bacteria. These plants can harbor or give the place to these bacteria to grow inside them. Okay, these plants form nodules. The nodules of these plants enlarge, and they give the place for these bacteria to live inside them. And inside the bacteria, if this is a bacteria, this is the leguminous plant, okay, and these are the roots of the leguminous plants. These plants form nodules, and inside the nodules they allow the bacteria, rhizobial bacteria, to live there, and in these places, that is inside the root nodules, these bacteria fix the biological nitrogen. Okay. Now when these plants die, what happens? These plants form a layer on the soil and they increase the fertility of the soil. Why? Because these plants helped in the fixation of nitrogen. So when they die, what happens? The nitrogen content of the soil increases, and thus the fertility of the soil increases. Okay. Now we should talk about the bacteria also. We have talked about the legume. Should talk about the bacteria also. So what bacteria are involved in this relation? These are azorhizobium, bradyrhizobium. So So Rhizobium is a kind of family which includes several Rhizobia species. Okay, Rhizobium, Cynorhizobium, and together they are grouped into Rhizobium. So what is Rhizobia? What is Rhizobium? So Rhizobium is a specific bacteria. So Rhizobia is a group which includes several Rhizobium bacteria, among which Rhizobium is a specific kind of bacteria. So this Rhizobia is a group. And Rhizobium, Azorhizobium, they are the specific kind of bacteria of Rhizobium. Okay. Now what is Rhizobium? What is the characteristic of Rhizobium? These Rhizobium are gram-negative bacteria, motile, they are rod-shaped bacteria, and they can either grow freely or they can live in the symbiotic association with the leguminous plant. Okay, so the existence of Rhizobium is not compulsory that they will live in symbiotic relationship with leguminous plant. They can live freely also, they can live with the leguminous plant also in the symbiotic relationship. Okay, it is not compulsory that they should live with the leguminous plant in the symbiotic association. Now coming with their association. So we have studied Rhizobium separately, we have studied legume separately. Now we will study about their association. Now what happens when any plant, like when the legume plant is infected with the Rhizobium bacteria, then what happens? Then these Rhizobium bacteria, wait, then these Rhizobium bacteria induce the plant to form root nodules. Okay, these Rhizobium bacteria induce the plant to form root nodules. What are root nodules? They are the specialized structure in the plants, in the leguminous plants, which will harbor this bacteria, and inside the root nodules only the bacteria will fix the nitrogen. Okay, so where inside the plant are these bacteria living? These bacteria are living in the root nodules of the plant. When are these root nodules formed? These root nodules are formed when the plant is infected with the bacteria. Before that, the plant do not have the nodules. They are formed after the infection of the plants with the bacteria. Okay, so how this process happens, how the nodule formation occurs? They are divided into two processes which occur simultaneously. So the one is infection and the second is nodule organogenesis. Okay, we will discuss it in detail in the next step. So look here at the picture. In the first step, what happens? So if it's an enlarged view of the root region, so what we see, this is a root hair. Okay, now, in the vicinity of the root hair, we can observe that there are several bacteria named Rhizobium. Okay. Now, these bacteria, see, large number of these bacteria are accumulated in the root hair region. In the B step, what we see that when these bacteria induce the curling of the root hair. So, first step was the bacteria will come closer to the root hair. How it will come closer, we will discuss. But giving you just the overview, the bacteria has come closer to the root hair. Now this bacteria had induced the curling of the root hair. Once the root hair has curled, the bacteria which are residing in this region, in the curled root hair region, in this region, they digest the plasma membrane in this region. They digest the plasma membrane in this region. As a result, what happens? As a result, what happens? Here, the plasma membrane breaks off. Okay, and sorry, the cell wall in the plasma membrane breaks off and the bacteria takes the entry into the root hair. After that, what happens? There is the formation of infection thread. That is, in this infection thread is formed by the fusion of the Golgi vesicles, and after that, then through the infection thread, the bacteria reaches the cortex of the root hair. Sorry, the cortex of the root. Now here, once they have reached the cortex of the root, what will happen? They have fused with the plasma membrane of the cortical cells. After that, they have branched into the several cells of the cortex. Okay. After that, while they were released into the cortical cells, you can observe that now they are covered or enclosed in a membrane known as peribacteroid membrane. So while they were released into the while they were released from the root hair cells into the cortical cells, they are enclosed in a membrane known as peribacteroid membrane. Okay. So to keep the summary, what happens first? The rhizobia comes in contact with the root hair of the host plant. This is the host plant, which is a leguminous plant. Here what we see that the curling occurred in response to rhizobia. The rhizobia digests the cellulose layer here and takes the entry. Now once they have taken the entry, they will form what? They will form the infection thread and takes the entry into the cortical cells. Once they have taken the entry into the cortical cells, what will happen? The infection thread will, when it passes to the cortical cells, they will fuse with the plasma membrane of the cortical cells. Now when they have fused with the plasma membrane of the cortical cells, what happens? They branch into several cortical cells. Once they branch into several cortical cells, there is a signal from the plant to stop the division now and to release the bacteria. The division or the growth of the infection thread will now stop and the bacteria will be released. So your free bacteria are not released, the naked bacteria are not released. The bacteria, when they are released into the respective cortical cells here and here, you can see they are now enclosed in a peribacteroid membrane. We will discuss about the peribacteroid membrane in detail. So see here the steps I have discussed. Bacteria first will come in contact with the root hair. Okay. After that the curling occurs. When the curling occurs, there is a degradation of root hair wall, which is a cell wall, and the infection thread is formed. How the elongated thread is formed? By the fusion of the Golgi vesicles of the root cells. After that, once it reaches the end of the cell, it fuses with the plasma membrane of the cortex. Okay. Now we have to understand about the mechanism, each and every step's mechanism in detail. So we have to understand that how bacteria come in contact with the nearby leguminous plant. So there is an exchange of the signals between them. So what kind of signals? So in the first stage, what happens? The bacteria migrates towards the root of the host plant. How they migrate? What gives them the signal that they should migrate to the leguminous plant? So this response is a chemotactic response because the chemicals are released from the leguminous plant. What kind of chemicals? The chemicals are isoflavonoids and betaines. Some phenolic signals are released from the leguminous plants which attracts the rhizobium towards the roots. Okay. Now these rhizobium have also a specific kind of protein named as rhicadhesin. Okay, rhicadhesin, which is present on the rhizobium species. So these are the rhizobium. These rhizobium species, the protein named as rhicadhesin, and leguminous plant also release some kind of attractant known as flavonoids. Okay, which are the phenolic attractants. Now these flavonoids, when they are released, what will happen? These bacteria which have rhicadhesin on their surface, they will be attracted to the flavonoids. Rhicadhesin are the surface proteins which helps in the attachment of the bacteria to the leguminous plants. Okay, this rhicadhesin is a calcium binding protein which play an important role in the attachment of the bacteria to the leguminous plants. So what we discussed, that a leguminous plant releases what? Leguminous plant releases phenolic compounds like flavonoids, betaines, homoserines, and these chemicals are sensed by the bacteria. These bacteria have rhicadhesin onto their surface, which helps in their attachment with the leguminous plants. So ricadison is a kind of attachment protein present in on the surface of the bacteria. Now, what happens once the attachment occurs? What happens? The bacteria will induce the formation of nodules in the plant. As I told earlier, that bacteria will induce the nodule formation in the plants. Okay, so we have to discuss about two genes. One is nodulin genes, which is specific to the plant genes. So plant genes specific to nodules are nodulin genes, and the rhizobial genes that participate in nodule formation are nodulation genes. So we have to study two genes, nodulin and nodulation genes. The host or the plant specific nodule genes are known as nodulin genes. Okay, the question comes from this topic. The plant specific nodule genes is nodulin gene, and the bacteria specific nodule gene is nodulation gene. Okay, now what happens? The legume plant secretes signals we already discussed. Now these signals can diffuse across the bacterial membrane, and bacteria, when they recognize these kind of signals, how they recognize these signals? They recognize these signals by the presence of a transcription factor inside them. Okay, these transcription factors inside the bacteria are encoded by NOD gene. Okay, now what happens when the root exudates? Say for example, these root exudates are releasing what? They are releasing flavonoids, they are releasing chemicals. These chemicals activate what? These chemicals activate the NOD D protein of whose? The NOD D protein of bacteria. Once the NOD D protein of bacteria is activated, these NOD D protein activates several other NOD genes. What several other NOD genes? The several other NOD genes are NOD A gene, NOD B gene, and NOD C gene. So what happens? Leguminous plants, the root of leguminous plants exudates or releases some chemicals. These chemicals activates the NOD D protein of bacteria. Once the NOD D protein is activated, they activate several other NOD genes which are NOD A, B, and C gene. So this part is of bacteria, and this part is of legume. Now what happens once the NOD D protein is activated? So NOD D gene is denoted by small, which will activate or which will produce a NOD D protein. NOD D protein, once it is formed, what will happen? This NOD D protein will bind to the NOD box. So there is a NOD box to rest of all the NOD genes. What are the rest of the NOD genes? A, B, C. So it binds to the NOD box of NOD A, B, and C. Okay, once NOD D protein binds to the NOD box of NOD A, B, and D gene, A, B, and C gene. So this dark one is the NOD D protein. So once NOD D protein binds to the NOD box of NOD A, B, and C gene, what happens? It helps in the formation of NOD A and NOD B and NOD C protein. Okay, so remember the NOD box is present only on the promoter of A, B, and C gene. The NOD box is not present on the promoter of NOD B. Now classification of genes. So there are two kinds of NOD genes, or we classify the NOD genes into two groups: common NOD genes and host specific NOD genes. The common NOD genes are A, B, and C. The NOD A, B, C are common ones which are found in all rhizobial strains. So we discussed about several rhizobial strains. There are A, B, C which are found in all rhizobial strains, but P, Q, H, F, and E. So 6 P, Q, H, F, E, and L, they differ among rhizobial species and they determine the host range. Okay. Now only one of the NOD gene, which is a regulatory NOD gene, Now we have to remember the regulatory NOD gene is a NOD D gene, which is constitutively expressed. It's expressed all the time, and its protein product, which is capital N NOD D, it regulates the transcription of all other NOD genes. Now what happens? Now these genes, which is the NOD D gene and the NOD A, B, C genes, they are involved in the biosynthesis and secretion of several other signaling molecules known as NOD factors. Okay, so three of the NOD genes we discussed, which are the common NOD genes, which are A, B, and C NOD genes. They encode the enzymes. So NOD A, NOD B, and NOD C proteins. The NOD A and NOD B and NOD D genes. This encode NOD D proteins. They encode the NOD factors. Okay, so these NOD, they encode the NOD A protein and the NOD B protein and NOD C proteins. They are together called as NOD factors. Okay, now these NOD factors or nodulation factors, what is the chemical nature of these NOD factors? They are lipopolysaccharide and they are the derivatives of chitin. So these are the derivatives of chitin. So these NOD factors, what is the chemical nature? They are beta-1,4 linked N-acetyl D-glucosamine. This is very important because question comes from you. Okay, they are beta-1,4 linked N-acetyl D-glucosamine. Now, as we said that different hosts have the host specific NOD genes are what? They are P, Q, H, F, E, and L. So what we see that how this P, Q, H, F, E, L are different, how they seem different, because the non-reducing terminal sugar moiety is substituted with a fatty acid. Okay, so the terminal sugar, which is non-reducing, this is substituted with a fatty acid, and the structure of this non-reducing terminal sugar moiety varies between different rhizobial species. Okay. Now what these NOD factors ultimately do, they help in the curling of the root hair cells. Okay, and they also helps in the cell division. Now during the infection, what happens? Rhizobia, which are attached to the root hairs, they release NOD factors. The rhizobia which are attached to the root hair cells, they release NOD factors which induce the curling of the root hair cells, and they also send mitotic signals. So what happens now? NOD factors, what do they do? The role of NOD factors is here. They helps in curling of the root hairs, and they also sends mitotic signals which helps in the cell division of the cell cortex. So what is the role of your cell division of the cell cortex? Which will help in the enlargement of the cortical cells to form the nodule. Okay, so cell division will cause the enlargement of the cortical cells to form the nodule. Okay, now these NOD factors inside the bacteria, they also induce several nodule specific plant genes. So first the NOD genes of the bacteria are activated. Okay, then they also activate the plant NOD genes named as nodulin genes. So see, now this is also important from where the question comes. Okay, so what is the chemical nature of this NOD gene? NOD gene is beta-1,4 linked N-acetyl D-glucosamine. Okay, now remember this is 1,4 linked. Okay, this is beta-1,4 linked. Now what is the role of NOD in the formation of this chemical structure? Formation of this beta-1,4 linked N-acetyl D-glucosamine, formation of this requires NOD A, B and C gene. Okay, so you have to just remember the function of each of those. NOD A is an N-acetyl transferase. It helps in the addition of fatty acid chain. Okay, now NOD B, what it does, it is a chitin oligosaccharide deacetylase. So one is acetylase transferase Another is deacetylase. Now deacetylase, as the name says, it removes acetyl group. So this is in total, these three are helping in the formation of this structure of the NOD factor. Okay. Now NOD C is what it is doing. It is a chitin oligosaccharide synthase that links N-acetyl D-glucosamine monomers. So this glucosamine is a polymer. Okay. So several monomers are linked by NOD C. Okay. Next. Now what we have seen that these NOD factors are first activated in the bacterial cells and they will help in the formation of NOD factor. Now these NOD factors will get attached to the specific proteins of the plants. Okay, so now I am talking about bacteria. These NOD factors from bacteria, they attach with the legume cells by a specific protein. If this is a legume cell, okay, they attach to the legume cell by a specific protein. So these are NOD factors. They attach to the legume cells by specific proteins named as lectins. Okay. Now NOD factors, when attached to the lectins are activated. Now this lectin directs particular rhizobia to appropriate host. Okay, and facilitates attachment of rhizobia to the cell wall of a root hair. Okay, so this lectin will activate and directs particular rhizobia to the appropriate host. Okay, this host range can be determined by this kind of attachment. Now once the NOD factors bind to lectins, which helps in the specification of the host of a particular rhizobia. Now what happens after the signal has been exchanged? What will happen? The curling occurs. After the curling, what happens? The rhizobia become enclosed in the small compartment formed by curling. We already discussed and now we are studying in detail. They are enclosed in a small compartment which are formed by curling. Now what happens? They degrade the cell wall in these regions, allowing the bacteria to enter, to cross the plasma membrane. After that, the infection thread is formed. Now what is infection thread? Again I am explaining. Now this infection thread is that bacteria migrates from the root hair towards the cortical cell. Is the infection thread. Okay. Now what happens? How they reaches downward by the fusion with the several Golgi vesicles near the site of the infection. Now the thread grows at its tip by the fusion of secretory vesicles to the end of the tube. It reaches to the end of the tube and reaches ultimately to where? To the cortical cells. Okay. Now what happens when the thread reaches specialized cells within the nodule? So nodule is what? Nodule is that when the cell cortex will divide and enlarge, it will form nodule. Once it reaches the nodule, what will happen? It will fuse with the plasma membrane of the host cell. Okay. Once they have reached into the plasma membrane of the host cell, what will happen? Now they have come to the cortical cells. Okay. They have come to the cortical cells. Here what will happen? They will be fused with the plasma membrane of the host cells. Now they will branch and they will grow further. Now there will be a signal from the host cell to not to divide anymore, not to grow anymore. When that signal is received, what will happen? Now they will not grow anymore, but the bacteria are now released into the cell. They will not grow, but the bacteria are released into the cell. And when bacteria are released into the cell, they are enclosed in the peribacteroid membrane. So inside here, when they are about to release, they are enclosed in the peribacteroid membrane. Okay, so they are enclosed inside the peribacteroid membrane. Okay. Now what we see, there is an undetermined signal from the plant which stops the bacteria to divide anymore and helps in the enlargement and uh, differentiate into nitrogen fixing endosymbiotic organelles called bacteroids. Okay, so the bacteroids are the enlarged form of the bacteria. Bacteroids are the enlarged form of bacteria of the rhizobia bacteria. Okay, these bacteria, when the signal is received, what happens? When the signal is received by the bacteria, what happens? They stop dividing and they become enlarged and they form what? Bacteroids. Okay, they form bacteroids. Just a moment. Now, these bacteria have a covering known as peribacteroid membrane. Now, these bacteroids have a covering known as peribacteroid membrane. This membrane, these are bacteroids, and this is a peribacteroid membrane. Okay, and inside this peribacteroid membrane, the nitrogen fixation occurs. Okay, inside this peribacteroid membrane, the nitrogen fixation occurs, and this peribacteroid membrane is derived from the host. This peribacteroid membrane is derived from the host. Okay. Now, in the peribacteroid membrane, what happens? Nitrogen fixation occurs. So, nitrogenase enzyme is involved, which helps in the fixation of nitrogen to ammonia. Okay, so this enzyme is very sensitive to oxygen. So, to control the level of oxygen here, there is a protein named as Leghaemoglobin which acts. Okay, so where is this Leghaemoglobin present? This is present in the cytoplasm of the infected nodule cells. It is not present in the peribacteroid membrane. Okay, it is present in the cytoplasm of the infected nodule cells at a very high concentration. So what this Leghaemoglobin does? This Leghaemoglobin transports all the all the oxygen. So bacteria, when they are living, these bacteria respire. When they respire, they release oxygen. So this oxygen is the presence of oxygen does not allow the nitrogen fixation. Okay, so leg hemoglobin what leg hemoglobin does is that once bacteria releases oxygen by respiration, they what they do? They removes the oxygen from the peribacteroid membrane. Okay, so see this in the leg hemoglobin part, the hem part is produced by the bacteria, and the globin part is present or produced by the host plant. Okay, globin part is produced by the host plant, and the hem part is produced by the bacteria, and it transports the hem part. Once it is formed by the bacteria, it is transported to the host cytoplasm where the formation of leg hemoglobin occurs. Okay, and this leg hemoglobin is pink in color. Now what it does, this leg hemoglobin has a high affinity for oxygen, for about 10 times higher than that of beta chain of human hemoglobin. Human hemoglobin also have a very high affinity for oxygen, but it was found that leg hemoglobin has about 10 times higher affinity than the human hemoglobin. Now what this leg hemoglobin does, it functions to transport oxygen to the respiring symbiotic bacterial cells in a manner analogous to hemoglobin. Okay, it does not allow the oxygen to remain in the peribacteroid membrane. It transports all the uh, oxygen to the respiring tissues in animals. Sorry, to the respiring symbiotic bacterial cells. This Leghaemoglobin transports all the oxygen to the respiring symbiotic bacterial cells. Now what happens? These nodules develop a vascular system. In the vascular system, what do? What is the role of formation of vascular system? First, that the nutrients are exchanged. Whatever nitrogen is fixed by the bacteria and whatever the raw materials are required for the fixing of the nitrogen, they are exchanged by this vascular system. Okay, and the oxygen which is produced is excluded from the root nodule interior. Okay, so that inside the interior of the root nodule where the nitrogen fixation occurs, that region should be devoid of oxygen. So they exclude the oxygen from the root nodule interior. Now this nodule, the nodules of temperate legumes are elongated and cylindrical, and the nodules of tropical legumes are spherical, just for the knowledge. Now, already I discussed that the presence of the symbiotic association is not obligatory. They come in the symbiotic association only when there is a requirement of the nitrogen. Okay, so rhizobia also occur as free living organisms, and legume also can live as unassociated throughout their life. Okay. Now what we see, that whenever there is a nitrogen limited condition, in that case only they come in contact with each other by the elaborate exchange of signals between them. Okay, the nitrogen limited condition. Now we have to study that there are some bacteria which form symbiotic association with the plants to fix nitrogen, and there are another kinds of bacteria which do not want any kind of association to fix the nitrogen. So these are free living bacteria, cyanobacteria, also known as blue-green algae. They fix the nitrogen without any association with any other plants. Green sulfur bacteria, azotobacter, and there are symbiotic bacteria like rhizobium which forms symbiotic association with leguminous plant, and spirillum which forms association with cereals. Okay. Now we'll solve some CSIR NET questions. So in June 2016, it came that rhizobial genes that participate in legume nodule formation are called nodulation genes. The NOD D encoded protein, we already discussed about NOD D encoded proteins. Okay, it is an acetyl transferase. NOD A was acetyl transferase. Okay, so this is wrong. So what we have to find? We have to find the correct answer. So this is wrong. Binds to NOD box and induces transcription of all NOD genes, we already discussed. Catalyzes the linkage of N-acetyl glucosamine residues. So this was the role of NOD C. It induces the host specificity of rhizobium. So host specificity were other genes. It was NOD P, Q, L, etc. Okay, so the B is the correct answer. Now, it came in December 2015 that the nodulation genes are classified as common nod genes and host-specific nod genes. So, we have to find out the common ones and the host-specific ones. So, nod A is a common one nod gene and nod C is a host-specific. We discussed that common ones are A, B and C. So, just find out from the simpler one that A is a common gene, yes, but C is not a host specific one, so this is wrong. Nod B is a common nod gene, correct, but nod P is a host specific, correct. Nod Q is a common nod gene, wrong. Nod P is a host specific, Q is a host specific, H is also host specific. So nod Q is a common, wrong. Nod H is common, wrong. So what we see, that B is the only correct answer. So I have given the explanation, you can go through it. The last question, symbiotic nitrogen fixation in legume nodules involves complex interaction between rhizobium and legume roots. This complex interaction is governed by, so integration of symplasmid of rhizobium in the root nuclear genome. We did not find throughout this class that there is any kind of integration involved. Okay, so there is no integration involved. This is just a chemical exchange of signals. Okay, without any integration. exchange of signals and there is no integration involved. Okay, sensing of plant flavonoids by rhizobia. Yes, right. Plant release the flavonoids and rhizobia sense it. Activation of nod genes. Yes, activation of nodulin genes. We discussed that the plant genes were known as nodulation genes. Sorry, the bacterial genes are known as nodulation genes and the legume genes are known as nodulin genes. Okay, so these were the kind of common questions which come from this topic. I hope this has helped you all. So for any query you can post it, you can ask me separately. Thank you.